Hi everyone, my name is Sarah and I chose to research the genetic disorder called cystic fibrosis. So let's begin with a little bit of a background on the disorder. Cystic fibrosis is also known as just the initial CF, and it is a hereditary disease that affects the lungs and the digestive system. Back in 1938, a pathologist by the name of Dorothy Anderson was the first to give a modern description of the disease, which she called cystic fibrosis of the pancreas due to autopsies of children that had died of malnutrition. Fast forward to 1963 when a guide for diagnosing and managing the disorder was produced by a panel of experts with the CF Foundation. Then in 1989, the CFTR gene responsible for the disease was first discovered and linked, which has made diagnosis a lot more easy to determine, and more recently, new gene therapies have been launched that specifically target the defective protein that generates cystic fibrosis. For the prevalence, CF is called a rare disease because there are fewer than 200,000 cases across the U.S. each year. It is more prevalent in Caucasian populations, with a prevalence rate being 1 in 2,500 to 3,500 white newborns. For other ethnic groups, it's a lot less common, affecting about 1 in 17,000 African Americans and 1 in about 31,000 Asian Americans. And CF is considered life-threatening, and people with the condition tend to have a shorter than normal lifespan. For the physical effects of the disorder, the thick mucus associated with cystic fibrosis often blocks passageways that carry air in and out of the lungs. This abnormal flow of mucus can also obstruct the channels that bring the enzymes produced by the pancreas to the small intestine. So the small intestine then is prevented from absorbing the nutrients from the food, resulting in many adverse effects. And cystic fibrosis can also affect the pancreas, liver, and other glandular organs. So some specific symptoms include whole body fatigue or the inability to exercise, a chronic cough associated with blood or phlegm, gastrointestinal issues such as diarrhea, heartburn, and a painful abdomen, respiratory effects such as pulmonary hypertension and shortness of breath, and delayed development or slow growth. So there actually is no cure for CF. Treatment then will depend on the severity and is largely focused on easing the symptoms and reducing any complications. As shown in that picture there, sometimes a chest wall oscillation device is used, which helps with removing mucus from the lungs by shaking the lungs and the mucus that would be on the walls. And medications then would include dietary supplements or antibiotics, and preventative medicine could include the pneumococcal vaccine. So what then would be the function of a normal non-mutated protein? Well, the CFTR gene gives instructions for producing a protein called the cystic fibrosis transmembrane conductance regulator. It's a mouthful. And this role of the protein is to act as a channel across the membrane of cells that produce sweat, mucus, saliva, digestive enzymes, and tears. This channel transports chloride ions, which are those negatively charged particles, out of the cells, which helps control the movement of water. That is responsible for adequate mucus production and flow, so that mucus helps protect the lining of organs and tissues by lubricating them, as well as the protein being responsible for maintaining normal function of other organs, such as the lungs, by maintaining other channels as well, such as sodium channels. So then, for the mutation and its impact, there have been over 1,000 mutations in the CFTR gene identified, the most common one being F508. The 508 is named for its location in the CFTR protein and the deletion of one of these amino acids at that location. That mutation in the CFTR gene alters the structure, the production, and the stability of the chloride channel, which then impairs the flow of chloride ions and the subsequent movement of water in and out of the cells, which then is going to lead to abnormal mucus production and consistency, therefore resulting in airway obstruction and a lack of proper lubrication on organs and tissues. So cystic fibrosis is called an autosomal recessive pattern mode of inheritance, meaning that mutations would need to be found in both copies of the gene in each cell. And an individual that carries a copy of the mutated gene doesn't necessarily show any symptoms of it. Again, you'd have to have both of those copies in order to present for the disease. So as shown here in this example pedigree, you can see that an individual with red in the corner would be someone that has cystic fibrosis, and under their name you can see that they would then have 2 copies of the little c, 2 copies of that recessive gene then. An individual with a light pink dot in the middle of their shape would be someone that is a carrier, so they got 1 copy of that lowercase c of the recessive gene and a dominant copy, or that capital C, so that they do not have the disorder but they carry it. And then someone with the blue in the center of their shape would be someone that received both copies of the dominant, so 2 of those capital C's. So they do not have the disease and they do not carry the disease either. So you can kind of see that flow there. And genetic testing is really important for this disorder. Early diagnosis shows that early diagnosis allows affected infants to begin therapeutic interventions immediately and has shown to improve height, weight, nutrition, cognitive and lung functioning, and a reduction in hospitalizations. And so some of the tests that are used are one that involves either a chloride sweat test or a swab from your cheek and has saliva or blood on it, which will test for 23 of the most common disease-causing gene mutations. Prenatal diagnostic testing is used to see whether an unborn baby has cystic fibrosis or is a carrier, and that is done through chorionic villus sampling, which is a tissue sample from the placenta, or amniocentesis, which is a sample from the amniotic fluid. And newborn screening is performed during a baby's first 2 to 3 days of life and is now mandatory for all newborns. And it is done by drops of blood from a heel prick, which are placed on a special card and analyzed in a lab. And here are my references. Thank you for listening.